Hi. For today's methods description, I'll be talking about all the protocols you need and the extra information necessary to perform a Western blot. Now, there's two different protocols that you'll need to follow to have a successful Western blot. And the first one is our Waldring Lab Protocols SDS-PAGE protocol. So with a protein sample of about 10 micrograms or so, which should be a good enough or excess amount of your protein of interest or protein extraction sample, um, where we should be able to detect your protein of interest. And typically, this can be anywhere from 10 to 20 microliters in a well of a gel that can hold around 30 microliters in total. So here we've prepared about half of that for the sample itself in the samples section. And in the following section, we just prepare each column, each lane that contains a ladder will have 5 microliters. So if you have multiple samples, you might want to have two ladders, so a total of 10 microliters of ladder be prepared if you're performing only one gel. Now, for a typical SDS-PAGE Western blot protocol in our group, we make two gels, one which we will Comassi stain to reveal the proteins that are present on the gel and in your sample that you've placed in each lane. And the second gel is used for the wet transfer protocol in Western blot that I'll describe in a few. So once you have combined all of the necessary things for your samples, you can briefly centrifuge them and then poke a hole on the top with a needle um, in just your samples. So now at this point, you will want to you'll want to have the heat block ready to go at about 100 degrees um, prior to having all that ready. And in just your samples, you'll poke that hole and place it on the heat block for about 12 minutes to denature the protein. And once that's done, you'll centrifuge your samples um, prior to loading them into the gel. Now, during that 12-minute time, this is where we'll go to start preparing our gel. For gel preparation, we use a Bis-Tris 4 to 20% pre-cast gel, which are located in bags filled with solution. In these plastic bags, you'll remove the gel and take out the sort of the gel casting comb by holding the gel in your hands with both hands on either side and pushing up and off so that the comb comes off of the gel, and also remove the sticky bottom part of the gel, the precast gel, so that you don't have any interference with the voltage going through. You will also, with this Bis-Tris 4 to 20% gel comes a Tris MOPS SDS-PAGE running buffer, which you can prepare by following the packets protocol. It's very simple. Add the packet contents and then a liter of water to be ready to use immediately, and can be reused multiple times, except for the part that will be put into the actual gel holder in the electrophoresis bin where you will perform the SDS-PAGE. Now, in the case where you might have only one gel, which is typically not for when we do Western blot, but if you are using only one gel, there are some precast uh, sort of not they aren't gels, but they are in the shape of the precast gels as a block to have something on either side of the gel holders so that you can fill it with the tris mops buffer without it falling out. And once you've assembled all of those contents, the two gels, the holder in the electrophoresis bin, you can then, once the 12 minutes is up and you've centrifuged your samples, you can load them into the gel. And this is done just like we would do in a DNA gel in terms of how we load it, but maybe a little easier because the lanes or the wells are typically much bigger. And then we can run that gel for 200 volts. And I find that you typically will find the best results between 30 and 40 minutes. Once that 40 minutes is up, in the meantime, at about 25 to 30 minutes in this electrophoresis, this is where I start to prepare the Western blot. And so the initial, we skip this initial part here with the SDS-PAGE. We go right down to wet transfer. And so this is where we'll be starting talking about Western blot. So in this 30 to 40 minute time, at about 25 minutes, I start to prepare everything for the wet transfer. So this includes getting the membrane activated. We use a PVDF membrane as opposed to a nitrocellulose membrane. And we activate it by first incubating with 100% methanol for 30 seconds, followed by washing with water, millicube water, for 1 minute, and then finally incubating that in the 1X transfer buffer for 5 minutes. Now, the 1X transfer buffer described above here can be prepared by adding this amount of water, transfer buffer, and methanol at a total of 1 liter. We typically buy the 10X transfer buffer as a pre-made jug of transfer buffer. So then, after this, once you start this 5-minute incubation in transfer buffer for the PVDF membrane, one thing to mention here is that this is typically done in the cap containers of a pipette tip box because it seems to be a pretty good size to have enough volume and cover the membrane or gels that we need to wash in between all of these steps. So in that container, we are incubating our membrane and transfer buffer, and then we are performing a wet transfer. So with a wet transfer, we sort of had to assemble this sandwich that you can see here with all these different components to enable the wet transfer to happen successfully. And so the transfer tank is where we put the transfer tank, we put our buffer soaked sponges and our filter papers, 2 on each side. So a total of 4 filter papers and anywhere from 2 to 4 sponges, where 1 or 2 will go on either side. And so we put these sponges and the filter papers in our transfer buffer to soak while we wait for the SDS-PAGE to finish. And at about this point, we usually have, once we've set all of this up, we've cleaned our scalpel and tweezers with ethanol and placed them on Kim wipes. We have prepared an area for opening the gels, and we've prepared all of our components in transfer buffer. We have about 5 or 6 minutes left before the gel is finished. And so once the gel is finished, we can take that back to our gel opening station and open up the gel with a screwdriver by just sort of pushing up against the edges on the top and bottom of both sides to have it dislodge from the other side of the gel. And how we do this is we typically have the longer side on the bottom. So that means the side where it's not being exposed, the side that is typically facing inwards on the gel when it's being placed into the gel box. And then we can take off that top part, which is the exposed part, where we also have the bits of the gel wells at the top. And at the bottom, we might typically see a some sort of front. Typically, this will be a very deep blue color, the color of the LDS, which is purple. And so we might see that at the bottom of our gel if we didn't run it for long enough for the front to go off. So if we try to imagine what this looks like, we might have some little well bits hanging off the front and this front at the end. We then cut those off with the scalpel, making more of a rectangular shaped gel, which we put to the side as we prepare our wet transfer sandwich. So the wet transfer sandwich holder, you'll have this white indicating the red part and the black indicating in the black here on the top. So so the goal here is the movement of charge from negative to positive as a force for driving the protein onto the membrane from the gel. And so how this works is we're going from black, the electrons are moving from black to white. And so that's why we'll have the gel on one side here and the membrane on the other side downstream of where the charge is moving as sort of the direction in which we transfer the gel components onto the membrane. And that includes the ladder as well. Here where you might want to use a pre-stained ladder as an indicator that you've transferred successfully. So we assemble first put down our soaked sponge, then our two filter papers on top of that. We can then, with our tweezers, lay, without touching with our gloves, our membrane onto the filter papers, avoiding bubbles, and then of course place the gel onto the membrane. Place the gel onto the membrane, and here placing it in a way where we're definitely covering the entire membrane and also avoiding bubbles between the membrane and the gel itself. We'll put another layer of buffer soaked papers on top of that. And from here, we'll take a 15 mil conical tube to roll lightly on the top of this to try to get any extra bubbles outside and create a tighter connection between the PVDF membrane and the gel itself. And on top of that, put our last buffer soaked sponge, close our sandwich, lock it in place, and place it in our tank to perform the wet transfer. Now we'll fill up the tank with the remaining amount of transfer buffer necessary to go up to the fill line. And then we'll also take a cold pack from the freezer to place in our electrophoresis transfer box when we set this up in the cold room on top of a magnetic stir bar. So in total in our box, we have our sandwich that we put in on the proper side where red is facing red and black is facing black. We'll put in a magnetic stir bar, an ice pack, and have that be filled up to the fill line with all of those things inside of the box. Now, once everything's in, we can close it up and run the transfer. For now, for successful transfer has typically taken 2 hours. So a minimum of 2 hours, but no more. But running an overnight transfer may be too long in the conditions that we have set where we're performing this in a cold room. So 2 hours gives optimal results, even for very large proteins above 150 kilodaltons, as seen by the comparison to the latter. We set this gel to about 299 milliamps, 2 hours, and run this for those 2 hours. Now, once that's done, we'll need to move into the blocking stage. Now, this is the point where we are adding a buffer that is able to interact with potential off-target interactions and allow us to have increased sensitivity for our protein of interest with the desired labeling strategy. And so here we describe a 10% non-fat milk with TBS buffer, a blocking buffer that you'll make. Now, this is a decent buffer to use, but if your labeling system involves biotin, non-fat milk will not be compatible with this since it will bind to biotin. And so because of that, we can also use a 3% BSA 1X TBS buffer. Now, this will typically look like 20 milliliters of 1X TBS and 600 milligrams of BSA. We will set that up and allow that to shake for about 20 minutes, at least 20 minutes prior to when we want to start using that buffer. But then we can talk about what we'll do prior to the blocking. So right after the wet transfer is finished, we would disassemble that that sandwich and place the membrane with the gel facing side up. Cover that in TBS, 1X TBS, and wash this 2 times. Wash this 2 times prior to the blocking incubation. So the TBS description of how we make that is here. We make a 10X TBS with 24 grams of tris and 88 grams of sodium chloride, adding to a total of 900 mils of water to that. Because then we'll want to use some tris HCl to adjust the pH down to about 7.6, leading to a final volume of about 1 liter once we've added the necessary amount of tris HCl and water. This typically comes out to about 60 additional grams of tris HCl in our TBS buffer to get to about 7.6. It's measured by pH strips. We'll then dilute that in 900 mils of water and 100 mil of 10X TBS to make 1X TBS, which we can then use for our washing steps. Now, we'll block that membrane for 1 hour at room temperature, shaking, in the described 20 mils of TBS with 600 milligrams of BSA. And after that, we, of course, dispose and wash again but now with TBS-T instead of TBS. Now, the difference between TBS and TBS-T, well, that would be the addition of tween. So like I described, our 1X TBS, we can make that with 100 mils of 10X TBS and 900 mils of water. But then we'll also add about 1 milliliter of tween 20 to that liter total to get our 0.1%, 0.1% um, tween 20 in our TBST. And so with the TBST, we wash 2 times after performing the blocking and one more additional time in regular TBS prior to primary incubation. Now, either during the blocking, near the end of the blocking incubation, or at the beginning of these washes, you should prepare an additional blocking buffer, but now these buffers will contain our primary and secondary antibodies. Because here I'm describing a fluorescence binders, fluorescence antibodies as the mode in which we image and detect our protein. You can also perform a chemiluminescent Western blot protocol, which this protocol here describes in a little bit more detail. And so because of that, I'll just talk more about the fluorescent side of our approach. So you can prepare your dilution. I would typically recommend following the manufacturer recommended dilution for Western blot using whatever antibody it is that you desire. But of course, in that same 3% BSA TBS blocking buffer, 1x TBS blocking buffer, we allow that to fully dissolve, add our primary buffer, and after, of course, all of those washes, we'll incubate in that primary buffer. Now, at this stage, this protocol has taken a decent amount of time to complete throughout the day. And so typically here at this point is where I incubate my primary antibody solution at 4 degrees overnight. Because at this point you'll probably be nearing the end of the day, and so I'll incubate that in 4 degrees rotating overnight until the next day. And so in the next day, which will be the second day, the last day of performing the Western blot protocol, you'll continue with your TBST washes, following a TBS wash, or prior to a TBS wash that'll lead into your secondary antibody incubation, prepared in the same way that we would prepare our primary antibody, putting the correct amount dilution into the 20 ml TBS plus 3% BSA blocking solution. Now, once we prepared our secondary solution with enough time prior to the secondary incubation, we can then pour our secondary solution onto our membrane and incubate that for an hour at room temperature shaking. Once this is done, we just have a couple more washes to do, about 4 more washes in TBST. And once you finish with that last wash, you can take that over to the ChemiDoc imager, or whatever imaging system is capable of detecting your fluorescent antibody. Now, the ChemiDoc that we have available to us is capable of detecting Lexaflor 647, 488, and a couple other fluorescent wavelengths, including some far infrared detection as well. Now, just to come back to the Comasi blue staining, now, if you remember, I talked about how we perform 2 gels when we do the Western blot protocol. And this is because one of those we want to Comasi stain to sort of see what the total protein looks like on that gel. And so with our brilliant blue Comassi stain, it's a very simple protocol where we just wash for 5 minutes after removing that gel. Then we incubate that gel in the Comassi blue stain for about 15 minutes. We'll wash again for 5 minutes. And this time we're washing with water for the first and the last time. So not TBS or transfer buffer or anything. We're washing in water for all the Comassi staining steps. So one wash for 5 minutes, one incubation in Comassi blue for 15 minutes, and then again one wash in water for 5 minutes. And then you can select the Comassi blue stain option on the ChemiDoc imager to look at your total protein contents in each lane. And so now you know how to perform a Western blot. Thank you.